Hello.
How are you?
I'm pretty good.
Great. Yeah. So, yeah. What did you have any updates or?
Not really. I'm trying to investigate how other people have studied tensegrities.
Yeah.
Yeah, I've got one paper
What do you mean?
about it. How to calculate their mechanics.
Oh, okay.
Because they're basically unstable.
Okay.
Because they're in tension. So I have one paper on it and a couple of math papers. So I'm just looking into it a bit because I'm like other people do this, but they use things like Mathematica programming. So I don't know.
So that's something you don't want to learn.
Well, I've used Mathematica. It's okay.
Yeah. I mean, yeah. Thanks. Good luck.
I just turned off my video feed because it seemed to be stalling things.
Yeah. Okay.
Yeah. So I'm good. I haven't done much. So.
Oh no. Okay. Well, that's good. Yeah. Thanks for giving the update. We have, of course, our two new GSOC students, Leywith and Jayadratha, and they've been to the meetings before. I don't know if they're going to be here today. We're in our community period, which means that they're getting up to speed on the community. We have the OpenWorm group, and then we have the Orthogonal Research and Education Lab group. And they've been kind of interacting more with our Orthogonal Lab group so far. We have our open source meetings, and we've been talking about different topics in open source, really kind of getting a baseline appreciation for the organizations and some aspects of open source. So I think in about 2 weeks they're going to start coding period, and that's where they're going to start producing demos and things like that based on their projects. So congratulations to Jayadratha and Leywith. And their projects are going to be DevoGraph projects. So DevoGraph projects, like basically we got like about 7 or 8 applications for that project, and so we'll have to maybe towards the end of the community period, I'll have them present on their project proposals and make sure that we know where they're going with that. Looks like Andreii is here, and Andreii is here. I'm just software development, which try to better understand what's in the OpenWorm group going in, and if I can help. Yeah, that welcome. Yeah, so we're the DevoWorm group. We're part of OpenWorm. We're kind of independent of OpenWorm in a lot of ways. And what we're interested in is developmental or computational developmental biology. So we're interested in developmental biology of the worm, of C. elegans. We're interested in like the embryo and modeling the embryo, modeling some of the developmental stages of the worm. There's a lot of embryo physics involved in that. There's a lot of data collection, like imaging technologies that we'll talk about. There's a lot of stuff like what Susan's doing, which is biotensegrity, which is looking at the mechanics of the worm, looking at how cells kind of fit together into a tissue or into a structure, how those mechanical changes impact the phenotype, the way it behaves, et cetera, et cetera. And then, we have other groups. We do things like we work with networks, we work with dynamical systems to describe things like development and behavior and things like that. And so, the goal of OpenWorm is to give this modeling of the worm, modeling of the model organism C. elegans from different perspectives. So there are different projects, some that focus on neurons, some that focus on the electrical activity of neurons, some that focus on whole body modeling and biophysics, and then we're interested in development. And we're also interested in connectomes and developmental connectomes. So one of the things you have with C. elegans is you have that that full connectome that we know, and we understand the connections and we understand the cells. And so that's of great advantage when we're looking at any sort of question regarding like a nervous system or a connectome. We can understand that nervous system on a cell-by-cell basis, and then we can understand some of the circuits that are involved in behavior. But we can also approach that from a developmental perspective where we have, where those cells get put into place, where the nerve fibers get put into place, and all of that. So that's what we're doing, Andreii. And, we do have all sorts of different projects. We're always in our meetings. That's kind of the glue of our group. We're always talking about different things that people are interested in. And it's really, it's kind of up to any one individual to like take the initiative on something. If they find something really interesting, they can drive it forward, present in the meetings, do a demo, or just kind of, see see if something works. See if there's some computational problem that you're interested in, or a developmental problem, or something in like OpenWorm, like one of the models that they use in OpenWorm as well. It'd be interesting. So welcome, and hopefully you find this useful. So we have Soham and Priyanka here as well. We have Jesse and Morgan and Susan and Andreii and myself. Okay, welcome. So like I said, we're interested in a lot of different things having to do with biology. I'm going to talk about something now that's a computational model. I guess it's related to biology, but we'll see. So this is a paper that I think is coming out at SIGGRAPH this year. This is inverse geometric locomotion. So this is a paper that, so in computer graphics and computer vision, people have been interested in simulating movement of things. So like when you want to model, say like clouds in the sky, you want to be able to find a hyper-realistic model of those clouds, like the depth, the shape, the texture, and then you want to be able to animate that. So you want to be able to find an efficient way to animate all of that. And you don't necessarily care what's inside of this thing. You just want to show a visual representation of it. Sometimes you want to be able to simulate, like you want to break things down into segments and simulate those segments. You want to have something that's lifelike, or something you would see in the real world. But you also want to have something that's computationally efficient, meaning that you have to be able to simulate it without any sort of lag. So it becomes a problem of kind of how to do that. And so people have proposed a lot of different techniques over the years. There's been a lot of math spilled, a lot of like computing power and cool animation spilled. Yeah, so this is inverse geometric locomotion, and this is this is the animation here, kind of the shorthand of what they're trying to do. So they have this sort of worm, and I don't want to call it a C. elegans or anything, but basically it's a worm that moves, in a way that it kind of moves laterally. It slithers, sort of like C. elegans. But the idea is that this worm is moving through this environment around these obstacles. And so they want to be able to capture the kinematics, or the movement dynamics of this movement. So if you had like an actual organism, say, there would be like this kinetics of movement, which is the energetics of movement, the energetics of the muscles, the forces being produced by the muscles. And you'd model the muscles. You'd model maybe molecular components of the muscles and other things like that. If you want the kinematics, however, you want to model the movement. So it's a lot more free in terms of how you can produce that. You don't need a hyper realistic model of what's going on inside the phenotype here. You just need to reproduce the movement relative to this body. So this rigid body or the soft body is going to move, and you want to be able to have it move around obstacles. You want it to be able to move around this space, but you want to understand how it's producing those movements, right? So it's like a dynamical systems problem where you have this system with a certain number of degrees of freedom, or the number of ways in which it can kind of change its shape or change its orientation as it's moving. And that sort of enables the kinematics. So there's some path that it needs to follow, but it only has a certain number of degrees of freedom. And so in the course of doing that, you're able to produce a more or less lifelike looking movement. And the only thing you're constrained by is sort of the energy requirements. And in this case, how much you can render in the graphics engine these movements. So that's kind of what this is. So this is in inverse means that they're solving the problem using an inverse solution, meaning they're solving it backwards from the equations and they're mapping it to this thing, these animations. So I'll just read the abstract here. The abstract reads, numerous tasks in robotics and character animation involve solving combinations of inverse kinematics and motion planning problems that require the precise design of pose sequences to achieve desired motion objectives. So these are the pose sequences here, where you have this object that's at these different orientations, and the degrees of freedom are in a certain configuration, and they have to move based on what those look like. So, however, accounting for the complex interplay between body deformations and resulting motion, especially through interactions with the environment, this poses significant challenges for the design of such pose sequences. So, if we think about like what they're doing in cybernetic in OpenWorm, what they're doing in cybernetic is they're modeling the biophysical aspects of the worm body. And the simulations that they run are usually embedded in this medium that they present. So they have to model the medium in which the worm is moving through. It could be like a gel. It could be like a colloid. It could be something that has a certain density, a certain viscosity, etc. We talked about low Reynolds number regimes a couple weeks ago, where we have the Reynolds number, which is a hydrodynamic parameter, and it basically describes the flow that something encounters, whether it be a cell or an organism. And so that Reynolds number, if it's low, means it's a laminar flow and there isn't a lot of turbulence. And so we can do this with hydrodynamics, we can do this with solids, we can do this with other types of materials. And the idea is that this worm is going to be moving through that material, and the properties of that surface or that medium are going to affect the body and its dynamics and how it can move. And so that's what they do in cybernetic, is they model that process, and they want to understand, that the worm being a soft-bodied organism and not a rigid-bodied robot or something like that, can sort of conform to some of the demands of that medium, but there's a limit, and it requires a certain amount of energy expenditure. It requires a certain amount of movement compensation. So you can run simulations in that kind of an environment where you model different types of media, and you can get information about what the worm might experience in that environment. So it tells you something about muscle physiology. It tells you something about the way that the body is configured and so forth. So that's a very high computational thing, like high cost computational thing, these biophysical simulations. And this is pretty high computational thing as well, because you have this, you have to model all this movement as a dynamical system. And so, this is what we're trying to do here. That they're trying to do is they're trying to make things look really compelling. And what they're doing in the biophysical simulations is they're trying to get precise measurements. So, anyways, back to the abstract. We propose a computational framework to address these challenges in scenarios where the motion of a deformable body is entirely determined by dynamic changes of its shape. Complementing recent methods on the forward problem, which is mapping shape sequences to global motion trajectories, we addressed the inverse problem of optimizing shape sequences to achieve user-defined motion objectives. We demonstrate the effectiveness of our method through a diverse set of examples, producing realistic shape sequences that result in desired motion trajectories. So they have these shape sequences, which are the changes in this phenotype. It has to go through this obstacle course, and then that results in these desired motion trajectories, meaning it has to conform to what we might see from, like, say, a worm in nature, instead of like just something that you've never seen before. Because it could produce those kind of movements, but we're not really interested in those. So there is this video here. This is just kind of their SIGGRAPH video. So this is inverse geometric locomotion. This is snake locomotion, where you see this snake moving around these obstacles. And it's producing something that's like a snake-like movement, right? I mean, it doesn't look like it's trying to walk, or it doesn't look like it's just kind of moving straight forward. It's making these kind of movements. And again, this is, I wouldn't say it's necessarily biologically realistic in the sense of like biophysical simulation. It's just kind of realistic looking. So this is a single scallop. So this is a, go back here. All right, so that's the worm. And then there's the single scallop versus two scallops on a stick. So these are marine invertebrates. These are their shells, and they open this shell up, and there's one, and then there are two that are going in opposite directions. They're connected by a stick. So they're just showing these movements and how realistic they can be. This is a turning snake, where this is where the snake is trying to find a path forward, using its degrees of freedom to achieve this. Okay. If we go further, we see that we could do that with the snake some more. We have a falling cat using matching user-defined semantics. So we have this cat, this point mass model of a cat that's falling, and it's falling onto a surface. This is hinged kinematic locomotor. So this is just kind of some sort of geometric shape that's like exhibiting some behavior. And it's kind of using its degrees of freedom to produce movements. This is the kinematic locomotor out of planes, so it's just kind of moving around. This is a spherical locomotor, so this is where you have a sphere that's operating under the same concept. And again, they're just showing off the method on these different models. This is nothing. Well, I guess you could maybe model a virus like that or something. And this is like maybe a cell surface where you have undulations on the surface. But the idea is that you have this triangular mesh that's kind of moving around. This is 12 Laplacian eigenmodes. So they're using different techniques. So again, this is it's a spherical mesh. We have 12 points on this example. And so we could go through this all day, I guess. Actually, it's 7 minutes long. This is a ray that's moving. So this is, it looks like a ray. This is Purcell's swimmer. So this is a point mass model that's swimming around, producing this trajectory from this middle segment, and then it's being graphed down here. So you can see it's following this attractor with its movements, and it's producing these rhythmic movements that have this sort of chaotic behavior, semi-chaotic behavior. Okay, so I think that's enough of that. I just wanted to point that out. And then, of course, they're using inverse methods here, which means that they're translating back from a mathematical model to these movements. They're trying to infer what these movements are from the mathematical model. So that was that paper. I thought that was very interesting in light of some of the things we talk about in the group. Do we have any questions about that?
Can you share the link as well for the paper?
Yeah, let me do that.
And thank you for the detailed description.
No problem. Okay, here we go. That's the link. Andrei, in response to what I had talked about before I presented the paper, he says, "Thank you. It was definitely helpful. I know that I have a lot of things to learn, so this looks like a good start. I definitely lack biology." A lot of people who join the group either lack like computational knowledge or biological knowledge. So that's one of the things we try to do in the group, is to merge those together. Okay, I should ask, does anyone want to give any more updates? Soham and Priyanka maybe have something they wanted to share, or maybe Jesse or Morgan.
I have just started, and actually I was in transition to an internship as well. So I think this week would be a little hectic for me. But yeah, I would definitely catch up from next week moving onward.
Okay, sounds good. Yeah. Right. Okay, so let's see. Andrei had another question. Since this group is relatively separated from OpenWorm, is there a place on GitHub where I can take a look at what's there? Okay, so yeah, we have a couple of places on GitHub. Let me share the links here. So we have two locations on GitHub, and I might as well go over those, as I think they'd be useful to people. So the first one is our DevoWorm repository. That's the first link. And I'm sharing my screen now, so you should be able to see this. And so this is our kind of legacy repository where we do a lot of our projects. We have a lot of repositories, some of which are, kind of not operative anymore, and some of them are, and we revisit them from time to time. So in this repository, we have our weekly meeting archive, which just basically goes over our different meetings that we've had. We have weekly meetings going back to like 2020. So if you're interested in different topics, you can look through that list and see if there's something that's you want to follow up on. There are different projects people have done, ranging from proteomics and informatics to different projects having to do with modeling biology, different packages for image segmentation, different packages for network reconstruction. I'd have to point you to a specific repository if you're interested in more. I can answer questions about what's in here, because there are a lot of repositories and forks, and it's kind of hard to follow, exactly what's going on here. But that's okay. That's the way it usually goes when you have a GitHub account for years and years. Some of these things are associated with papers like this raising the worm brain. I'm looking for a link to our data sets. this is where it is, DevoWorm. So in GitHub, DevoWorm/DevoWorm, we have a bunch of data sets. And these are data sets that we've collected over the years. These are kind of like our baseline data sets. So if we're looking to train a model, or we're looking to build, some kind of data set, we might use some of these resources. So we have, of course, the C. elegans data from development. We have information about cell birth and death timing. I don't think we have the connectome data in this repository. It's in another place, but we have data from ascidians, or an organism called sea squirt. We have gene expression data, lineage tree data, and so forth. So there are a lot of data sets in this repository, DevoWorm. Then if you go back to, there should be another one for connectomes here. I'm not really putting my fingers on it right now, but we have another set of data for developmental connectomes, and that is, I'm not really sure what that is. But if you go through this, you'll find, a lot of repositories, and if it looks like something you're interested in, my suggestion for this would be to go through maybe the meetings first. Well, this is it. And think about what you're interested in, and then look for something in the GitHub repository. This is what I was looking for with the embryogenetic connectome. This has data for different stages in development and the cells that are involved in the connectome. This is linked to some work we've done on this topic. So there are CSV files in here, which are, they should give you the comma delimited format. There are other types of GraphML structures in here. So there, we have all sorts of different, we use all sorts of different programs to do these things. You have like graph theoretic programs, you have biophysical modeling programs. Susan does a lot of stuff with like cell mechanics. And then we build, and I'm going to show you that in a minute, we build our own software for image processing and building biological networks. So this is a data set for connectomes. So there are all sorts of data sets in here. The next one is DevoLearn, and that is this repository. Here's this organization. So in about 2020, we've kind of got to the point where we wanted to build a software platform for, segmenting images. So a lot of the biology that we do is like taking microscopy images, which you can get freely available microscopy data from different repositories. And you can get those data, but you can't necessarily say anything about the biology until you segment out things from the images. So if you have an image of like an embryo, you can't say much about that embryo until you've segmented the image into cells, into membranes, into nuclei, and have a map basically of what those structures are in different images. So if we have a time lapse of an embryo, we have cells, we have nuclei, sometimes cells are dividing, so we might have more cells in when we compare two different images, we might have more cells in that new image. We might have changes in the membrane. And those are the things we're really interested in. So we developed some software for this. It's ranged from like using sort of RNN models and pre-trained models to the segment anything model that Meta put out a couple years ago. And so we have a number of models for image processing. Those are located both on GitHub and in Hugging Face. If you go through DevoLearn, which is the platform, you'll see that we have a number of different models that we use here that are sort of at the heart of what we're doing with image processing. So we have a nucleus segmenter, a cell segmenter, and I think a semantic model. So this is really interesting stuff. And again, I can kind of help you navigate this if you're interested in something specific. And then, of course, we have DevoGraph, and this is the thing we've been working on more recently. This is, applying graph neural networks to these kind of microscopy images. So it's not enough just to segment the cells, although it's necessary to segment things into membranes and nuclei and other things like that. But then you want to know how they're related to one another. So that's where DevoGraph comes into play. So we have these graph structures that we can reconstruct from images. We segment the images, and then we take those components that we've segmented out, and we try to find the relationships between them using graph theory. And then we have graph neural networks, which process those graph structures and turn them into embeddings. And then we can say things about the biology, and we can make, hypotheses, but we have everything saved in a computational data structure. And so this is what our current Google Summer Code project is for this year. We've had a number of contributors to this over the years as well, so it's a pretty robust attempt at this. And again, that's maybe something you're interested in or not, but there are always these opportunities that come up to do things. So that's a little tour of our, and this is also for our GSOC students, because I want to make sure that they know what we have in terms of repositories and what we have in terms of resources available. I know that the GSOC students are very familiar with the DevoGraph repository, but maybe not so much with DevoLearn. That's been a focus of Google Summer Code projects in years past. So, but we haven't talked about it this year. So if you want to do something like cell segmentation, if you want to incorporate that into a project, I would prefer we draw from what we've done with DevoLearn instead of picking something else up off another shelf and applying that to the problem. But this is all stuff we need to talk about, though. And this is always a thing in open source where people will, do something in a repository, and it's kind of half-baked because they don't really know what's going on in the repository. And if you do that enough times, your project becomes really misaligned, and it's hard to deal with. So yeah, that's all we have in there. Whatever people find interesting, please let us know. So Susan is really interested, of course, like I said, in cellular mechanics. And we've been talking about this tricellular junctions topic. And so tricellular junctions are where you have a sheet of epithelial cells, and then there's a place where maybe 3 cell membranes will meet. And then that's considered a junction between different cells. A tricellular junction, of course, is 3 of those cells coming together at a point. You can have, I guess 4 junctions, more, 6 junctions, depending on the geometry and the packing of those cells. So that's those are, N cellular junctions, I guess, or N junctions. And she sent a couple papers along. So this is a paper, Tricellular Junctions. And I think we talked about this in a previous meeting, but it's worth bringing up again because I think it's, an interesting paper, especially given the topic. So I'm going to talk about this paper and then another paper she sent me. So this is Tricellular Junctions, how to build junctions at the trickiest points of epithelial cells. And TRIC is the acronym here. So it's very cute. And so the idea is that they're, these places that cells come together. And obviously, when you have a packing of cells, it's important to have places with great structural integrity, because that sheet of cells then bends and, has to bear loads and has to do things in the body that require this sort of structure. And of course, Susan's interested in biotensegrity, which is how to achieve ultra stability in these structures. So the end junctions are the places where you get this kind of, you could get this kind of ultra stability. And so then the abstract of this paper reads, tricellular contacts are the places where three cells meet. All right, that's where you have this geometry where they kind of come together, they get packed together into a point. Invertebrate epithelial cells, which are these kind of cells that are kind of like skin cells or other types of cells. They're usually kind of cubical. They have a certain shape, typical shape. They are found across vertebrates, and they form these sheets that are involved in like organogenesis and things like skin. Invertebrate epithelial cells, specialized structures called tricellular tight junctions or small t TJs, and tricellular adherens junctions or small t AJs. So we have tight junctions and adherens junctions, and those are just different types of junctions in the anatomy. They've been identified. TTJs are important for the maintenance of barrier function, and disruption of TTJ proteins contributes to familial deafness. TAJs have recently been attracting the attention of mechanobiologists because these sites are hotspots of epithelial tension. So when they say epithelial tension, they mean that the sheet of epithelial cells as a sheet, not just as the individual cells, but the collective behavior of those cells, form this sheet. And the sheet has its own sort of physics. So you have tension, you have compression, you have all these different properties of the sheet. And then, so they're basically saying that these are hotspots of tension. So again, ultra stability or tensegrity relies on these kind of emergent features of these larger scale structures. And then that's, of course, one of them. Although the molecular components, regulation, and function of TTJs and TAJs, as well as of invertebrate tricellular junctions, are beginning to be characterized, many questions remain. Here we broadly cover what is known about tricellular junctions, propose a new model for tension transmission at TAJs, and discuss key open questions. So this is where they were going in this paper. They talk about these different types of junctions, and so that we have the sort of the microscopy, which describes the morphology, and then we have this molecular aspect, which is where we observe these different proteins, and the function is kind of observe the function almost kind of semi-independently from the morphological structure. So we look at the morphological structure at the microscopy, and then we have the molecular aspects of it. The molecular components, sometimes you measure them sort of in situ and you get a good characterization of how they operate, but more often than not, we're doing a lot of annotation, we're doing a lot of inference of function. And so that, this is a problem sort of with bioinformatics, is that you have molecular data and we know kind of what things do, but it's very general characterization. A lot of times you need to do studies where you can actually observe like proteins or other types of things like gene expression at the site. And, our technologies to do that are kind of limited. So it's hard sometimes to match what's going on in the molecular milieu or the molecular world of like trijunctions with the morphology. And so that's, so this is actually where we can kind of get a connection between the two here. We have expression of angulin 1. We have the sort of the membranes here, and then the merge, where the angulin 1 is expressed at the junctions for the membrane. So these membranes show this these junctions. So here we have a trijunction, an example here, another example of a trijunction, where 3 cells come together. And as you'll notice, they have this kind of shape. This is, I guess, looking at the top of the sheet. So they have this shape that, they kind of squeeze into this conformation. And then you get these different trijunctions that form. So it's just basically wherever you have 3 cells coming together. And as you can see, you have a lot of edges to this. You have 1, 2, 3, 4, 5, 6. It's kind of a 6 or 7. 7. So it's like a 7-sided object that's kind of squeezed together.
Yeah, this is cell packing.
Cell packing, yeah. There's 7 trijunctions in that for this space. And so this is where the cells pack together very tightly and form these junctions. Now, that being said, the junctions are always changing. So the cells are migrating past one another. They're kind of, changing their shape. And so these trijunctions, sometimes they're stable, sometimes they move around. And I guess it's this expression of angulin 1 that really kind of defines where they are. And so that's an example of a study where you can get this expression of these angulin proteins and, how they play a role in function, or how you can infer that role. So I remember going through this paper, we talked about the molecular aspects, we talked about the anatomy. They have some nice figures in here showing kind of the connection between the two. So I remember this where you have the different tight junctions and adherence junctions. I guess if you go down, so this is looking at the top of the sheet, and then of course there's the dimension where you're going down. Looking at it from that perspective, you see that they're sort of cubical, they kind of go down, straight down like this. And that's an artifact of this model, but basically they have a depth as well. And so in that depth, we have along these junctions, we have all sorts of stuff going on along them. So those junctions aren't just points. They're actually like axes that go down the sides of these cells. And so you get all this expression of these different junctions here. And so that's kind of what we're looking at here. So I remember this paper was pretty good. It had a lot of nice illustrations. It talked about the importance of these junctions and how they play a role in the morphology of these cell sheets. This is the paper I think that Susan sent me more recently. And this is Tricellular secures the epithelial barrier at tricellular junctions by interacting with actomyosin. So the first paper was in Molecular Biology of the Cell, I believe, and this is from Journal of Cell Biology. Not that it necessarily matters, but this is more sort of, I think, a cell biology paper. So this paper says, or the abstract reads, the epithelial cell sheet functions as a barrier to prevent invasion of pathogens. So this is why we have epithelial cell sheets, maybe one reason. And so as a barrier, it needs to have some sort of stability. It needs to be, strong and, in the face of cell migration, in the face of maybe all sorts of invading things that come into the cell or into the tissue, I guess. It is necessary to eliminate intercellular gaps, not only at bicellular junctions, but also at tricellular contacts. So we have these bicellular junctions and these tricellular contacts where 3 cells meet to maintain epithelial barrier function. So you have to think in terms of the junctions, which are these proteins that connect, and then the tricellular contacts, which are where the physical aspects of the cells connect. To that end, tight junctions between adjacent cells. So you have these tight junctions in each cell, and they kind of, join together between adjacent cells. These tight junctions must associate as closely as possible, particularly at tricellular contacts. Tricellular is an integral component of tricellular tight junctions, or TTJs, which we talked about in the last paper. But the molecular mechanism of its contribution to the epithelial barrier function remains unclear. So, we don't necessarily know what the molecular mechanism is. We know that they exist. We know that these tight junctions exist. We know they need to be in a certain configuration. But it's hard, it's harder to infer a mechanism, especially in terms of causality and what it's doing. In this study, we revealed that tricellular contributes to barrier formation by regulating actomyosin organization at tricellular junctions. So this is where this tricellular is contributing to this barrier formation function of the sheet by regulating actomyosin organization. So actomyosin is in the cell. It's kind of enabling the movement of cells, the migration of cells against one another, and it allows for these tricellular junctions to remain stable despite a lot of changes in this sheet, a lot of stresses on the sheet, a lot of cell migration and things like that. Furthermore, we identify alpha-catenin, which is thought to function not only at adherence junctions. As a novel binding partner of tricellular, alpha-catenin bridges tricellular attachment to the bicellular actin cables that are anchored end-on at tricellular junctions. So we have these actin cables that we have actin within cell membranes. They sometimes have to span cell membranes. Actin, of course, is kind of like serves the role of kind of like a stretchy filament, and they're necessary for this sort of flexibility. And they need to be coordinated, because you need to remember, you don't need to maintain just the stability of a cell, but all these cells sort of in the array, and you want to maintain the stability of that array. And so they're all anchored at this tricellular junction. They're all coordinated at that point, and you don't want them to be uncoordinated. Right. Thus, tricellular mobilizes actomyosin contractility to close the lateral gap between the TJ strands of the three proximate cells that converge on tricellular junctions. So it's this, they're proposing this tricellular, which is the thing that's sort of mobilizing actomyosin contractility. So actomyosin acts a lot like muscle or skeletal muscle, where it contracts, and that allows you to move, and it allows a cell to move as well. So this is why it's important, because you get a lot of stresses, a lot of force loading on these sheets, and you want to maintain these sort of configurations as much as possible. You can imagine if all the cells were sort of they were sort of rigid bodies, and they weren't coordinated like this, that if something were to there were a stress put on the sheet would pretty easily collapse, and it would have to reassemble itself all the time. So this is one of the benefits of what we're talking about here. So again, they talk about tight junctions. They talk about these other types of junctions. Let me see if we have anything interesting here. So they talk about tricellular junctions being tension hotspots, or mechanical forces applied to cell adhesion molecules. Tricellular junctions experience high tension due to outward forces generated by actomyosin-dependent line tension acting along each bicellular junction. Homologs of neither tricellular nor LSR have been identified in invertebrate epithelial cells. So this is a vertebrate-specific thing. So in invertebrate epithelial cells, we have different means of maintaining these sheets. We talked last week, I think, or last meeting, about, these transitions in evolution where you get certain things that a whole bunch of things that happen at once that enable new structures. And so we talked about multicellularity in eukaryotic cells. And you see a lot of these things with like differences between invertebrates and vertebrates. And so, that, and of course, there's a lot of diversity in both taxonomic groups, but you can see that there are differences in how invertebrates have solved problems versus how vertebrates have solved problems. And sometimes you have things that are enabled in one group or another that are, important for the way their tissues form, the way they live their life. So it's, a lot of times we'll point to these differences between vertebrates and invertebrates. And it's interesting. Sometimes it's just that there's a difference in how maybe their common ancestor did something, or maybe it's an innovation that is unique to vertebrates. So it's always interesting to see these differences here. So, of course, when we're interested in C. elegans, we're interested in invertebrates. The model organism Drosophila as well as an invertebrate. So they kind of talk about invertebrates here for a while. And their differences, so some of them involve homologs and identifying those homologs. And these are just homologs of the different proteins. So maybe it's a different protein, but it's basically doing the same thing. So you have, but even in invertebrates, you have several cell adhesion molecules being enriched at these tricellular junctions. Among them is a protein called sidekick, which belongs to the immunoglobulin superfamily. Those are concentrated at tricellular adherence junctions in Drosophila. So in Drosophila, you have a system where it's a similar sort of morphology, but you have different molecules mediating those junctions. In a departure from the vertebrate system, sidekick itself mediates homophilic adhesions between the three cells that comprise the TAJ. Additionally, sidekick anchors the actomyosin cytoskeleton through polyketoid, which is the mammalian homolog of ZO1, and canoe, which is the mammalian homolog of afidin at tricellular junctions. So when they talk about these homologs, they're just like basically very similar proteins. They just have different names, but they do basically the same thing. They have, I think, the same evolutionary origin. However, in the case of vertebrate epithelia, the functional relationship between either tricellulin or LSR and the underlying actomyosin cytoskeleton at tricellular junctions is not known. So in the present study, we reveal that tricellulin anchors F-actin connected to tricellular junctions in an end-on manner via alpha-catenin. So we anchor F-actin, these tricellular junctions using tricellulin, and this is, done in an end-on manner, and alpha-catenin is mediating this. And alpha-catenin is the principal scaffold protein of AJs. Tricellulin plays an essential role in the formation of TTJs by coupling close association of two central sealing elements with actomyosin contractility at tricellular junctions. So this is not just about maintaining that junction, maintaining the cornering aspect of this, which maintains the stability of the sheet, but also it allows for this contractility of actomyosin, and it allows for the stability of the sheet in the face of that contractility. Because remember, you have three cells, and they're all contracting, and they may or may not be coordinated. So that's why we have all of this molecular machinery in place. Okay, so, and then they give some results here. From these results, we propose that the formation of the actin meshwork at tricellular junctions, so there's this meshwork of actin, is a distinct morphological stage during the maturation of epithelial junctions. In the following experiments, we examine the functional roles of the tricellular actin meshwork in the formation of epithelial barriers. So that's basically what they're trying to do. They're trying to understand these barriers. They're trying to understand how we can maintain these barriers through this sheet of epithelial cells, how to maintain stability of that sheet, both during like loads placed on it or stresses placed on it, and during like the movement of cells and the adjustment of cells and the contractility of actin in response to different stimuli. So there are a lot of things going on here. But I think it's an interesting set of papers because it does describe kind of, you know, what happens in organogenesis, right? You get these sheets of epithelial cells, and then they have to be sort of functionally robust. They serve different functions, and, you know, and there are a lot of things that are going on in the cells that are keeping them sort of in this association. And then that leads to, you know, further organogenesis. So, Susan, I know these are this is your kind of your area. So, would you like to speak to some of this?
It's interesting that cells are attached just through that junction, mainly through that junction. The bijunction is more a surface junction, and then the deeper junction is that trijunction. And it matters how wide that is in my modeling as how the tissue behaves. So cells are very reliant on their attachments. And the trijunction is the main one. And then the other one is the basement membrane attachments. So that's all important, and it gives a tissue is stronger than its individual cells by a lot. I have a paper here that I think it's at least 100-fold stronger mechanically than a single cell. Anyway, it has different characteristics when they're attached to each other. So studying individual cells doesn't give you a complete overview of what's going on in the body. You need to look at the tissue and the attachments.
Yeah.
That's just a sort of general thing.
Yeah. And then, you know, what do you think the broader role is for like tissue morphogenesis? I mean, what kinds of things? I mean, you know, in your thesis, did you think about, or did you kind of consider?
I just said it's a total thing. It's in general physiology, homeostasis. It's in cancer development, and it's in development, embryo development. Epithelial tissue and their attachments play important roles in all of those. Yeah, I don't know. I have another paper. I think Dick sent it to me. And it talks about that. So I'll send you that paper. I was reading it and I put it somewhere. Yeah, total organization here. Yeah, I don't know where I put it at the moment. Sorry.
That's okay. No, I think that's great. Yeah, I like that. You know, again, I can't remember what meeting it was, but it was a while back, and we did a couple papers, and then, you know, we keep coming up with new papers on this topic. I like the topic. It's interesting, you know, and this is a hot area because people are publishing in it and discovering new things all the time. So
yeah, it's neat that they've managed to figure out what a trijunction is made of, what proteins, and that's new. Like, that's a new development because of new microscopy,
Yeah.
new analysis. Yeah. It's a new area for sure.
Yeah. All right. Oh, I think that's all for today. It looks like Priyanka had to leave, but thank you for attending. And Soham, thank you. And then Andreii, thank you for attending. Hope you found this useful. And all right, well, see you next week. Thanks for attending. Have a good week.
Okay, bye. I'll send you that paper.
Okay. All right, thanks.